I'm sorry, that's not what I meant to say. What I meant to say was deoxyribose and ribose differ from each other, in the sense that in DNA, deoxyribose serves as the DNA's pentose, which is a 5-carbon sugar. And then in RNA, we have ribose serving as its particular pentose. And that pentose structure represents the sugar backbone of the DNA or RNA molecule. So in this figure here, the sugar molecule is located here and is affixed to a phosphate group. And the sugar molecule is affixed to a nitrogenous base, which makes up the nucleotide of our DNA or RNA. So for a DNA, which exists as double strands, you're going to see a duplication of that deoxyribose. And then in between the DNA, the latter ring portions of the DNA, you have your nucleotides which bind together. And in DNA, we have 4 primary nucleotides, which consist of adenine, thymine, guanine, and cytosine. So A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Now, these nitrogenous bases or nucleotides are bound together through the formation of hydrogen bonds, and the share pair or the share pairing between adenine and thymine that exists will only have 2 hydrogen bonds holding it together. When guanine binds to cytosine, it will have 3 hydrogen bonds holding it in place. And so if we needed to break the bonds between adenine and thymine or between guanine and cytosine, it would take more energy to break 3 hydrogen bonds than it would to break down 2 hydrogen bonds. And just kind of as an interesting note, when scientists look to see which particular genes have been conserved among the lineages of different species of animals, they'll look for the G to C bonds, because these bonds take much more energy to break, so the more conserved they are, the less evolution we're able to see. And we kind of look for those patterns to see, you know, which species evolved from which other species when we're studying the concept of evolution. But these pairings help to generate the double alpha helix that makes up our DNA. And as they're bound together, you start to get the spiraling because of the way that they are bound together. Having 2 double-strand polymers which exist on the side, and then these hydrogen bonds that hold these nucleotides in place, that helps to create the twisting force that you see whenever you think of a structure of DNA. And here's an image to kind of get out of the text, the slides that have text with them that I don't think really fully convey what, you know, a genetic molecule looks like. But again, here basically is a strand of DNA. We have our nucleotide bases, which exist as the rings of this DNA structure. So you have cytosine, guanine, thymine, adenine. Cytosine, as I said, has 3 hydrogen bonds with guanine. Thymine has 2 hydrogen bonds to adenine. So the 3 bonds represents an area where more energy is needed to break apart this bond sharing here. Now, all these nucleotides are tied or bound to the pentose, which can, in RNA is ribose, but in DNA, which this is DNA, we simply have deoxyribose. So you have a deoxyribose molecule that makes up the backbone of the latter. And remember, these backbones are held together via the phosphate groups, and so that's what these little circular structures on the side of the pentose molecules reflect. And again, remember that DNA is double-stranded. So we have 1 strand here of the deoxyribose bound to a phosphate group, which is connected to our nucleotides, and then we have a whole other strand that's also bound to nucleotides as well. And so this gives us the basic shape of what our genetic material looks like. And here's the molecular structure outlining that material. So Here is just another image showing you what this looks like. So we've looked at essentially the molecular structure, and then another schematogram kind of highlighting where the nucleotides are, and then this structure here, which is the more common form of what we tend to view whenever we think about genetic material, with your nucleotide bases in the middle, and then you have your deoxyribose phosphate backbone, also commonly known as a sugar phosphate backbone, that creates the structure and support needed to hold these nucleotides in place. And we also refer to this again as a double helix. Okay, so why is this important? Well, it's important for us to have genetic material so that way we can pass on our genes. So also it enables us to produce proteins inside of the cells, and we can also produce RNA in the cell. So all those structures are needed to help enable life for the cell. So for that to take place, we have to have the process of DNA replication, where what happens is a molecule of DNA is taken inside of the nucleus in our cells, and it starts to separate through the help or assistance of enzymes. And one of the most important enzymes that's involved in this process is the enzyme called helicase, which will help to break the bonds that exist between the rings of the ladder. So it will break the hydrogen bonds between adenine, thymine, guanine, and cytosine. When those bonds are broken, the two strands of DNA will start to unwind. And as they unwind, you have another enzyme called DNA polymerase, which helps to assist in the replication portion of this process. So DNA replication will take place when the DNA strands are separated, and once they're separated, DNA polymerase will go to a site on where DNA replication is occurring, and it will start to lay down new nucleotides. Okay, so let me try to increase this image a little bit so you can see this. So the old strand in this image is going to be the purple strand, and then the red strand is the newly formulated strand. So let's see where this process is starting. So it looks like the process here is starting with maybe where guanine is, and what you see here is that you have the laying down of new nucleotide pairs. Okay, so as this becomes open, you have thymine that isn't paired with anything, and adenine here that isn't paired with anything, and those go all the way down to this guanine that should be paired with cytosine, but it isn't. This guanine should be paired with cytosine, but again it is not. And then you have thymine that should be paired with adenine. So either way, we see that we have the split in the fork here. So what DNA polymerase will do is it's going to come through here, and it's going to lay down new nucleotide bases or generate them. And so it will start to build a nucleotide adenine to bind with thymine, a nucleotide cytosine to bind with guanine, guanine to bind with cytosine, guanine to bind with cytosine, thymine to bind with adenine, and adenine to bind with thymine. And then it will do the same on this portion of the strand template. And so let's take a look at this DNA replication video that was posted on YouTube by Your Genome to kind of watch to see this process unfold. So we have our DNA structure. And then what happens is you have an enzyme that comes in and helps to unravel the twisting structure of the DNA. So as that happens, you start to have a breaking apart of the DNA structure. Right now this video is outlining how the different strands are identified via 5' end and a 3' end, and then 3' to 5' end. So what's happening now is that this enzyme known as helicase Let me pause this real quick. Helicase is breaking apart the hydrogen bonds that exist between the nucleotide bases to separate them. So as the enzyme works its way down the DNA, you see that we have the formation of what's called a replication fork. So here's another video kind of outlining what happens when we have the process of replication. There's the helicase we talked about.
Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
So I'm not expecting you to memorize all of the different components that are involved in the process of replication, but just know that we use these biomolecules pretty extensively to help develop, help enable this process to take place. Okay, so of the enzymes that we saw, the two primary ones are helicase, which help to break apart the double helix, and then polymerase, which helps to put down new nucleotides. The process of DNA replication, like you saw in the video, is called semi-conservative, which means essentially that you have one region that contains, or one strand that's going to contain the same genetic material, but the other strand that is red isn't going to be conservative. So the new versus the old is what we mean when we talk about semi-conservative. So half of the genetic material is new and half is conservative, or exactly the same as the parental DNA strands. So in the idea of gene expression, just so you know, a gene is nothing more than a segment of a DNA molecule or a chromosome. So where you see all of these guanine to cytosine, thymines binded to adenine, all of those, that little segment right there can be considered a gene, because a gene just has information in it that can be copied, it can be translated and then transcribed, or transcribed and then translated into a protein for whatever specific function. And when that happens, we refer to what's being produced as a gene product. Okay, so genes are real important because they're codes that give us different products that can be used by the cell. And as I've said, for these genes consist of adenine, guanine, cytosine, and thymine. So the order in which these are paired together will make up what genes are present inside of the DNA. Most genes are all going to code for proteins, but there's a lot of genes inside the genetic code that we don't know what they do. Other attributes that these genes can code for would be things like ribosomal RNA or transfer RNA, both which are used in the process of transcription and translation. Okay, so with DNA replication, there's a particular theory that is it's a dogma theory, the central dogma theory, that is used to describe how genetic code is converted into a protein to be used by a living organism. So the central dogma theory basically says DNA is transcribed into messenger RNA. So what happens in this process is that you have that deoxynucleic acid being converted into a new molecular structure, which is known as messenger ribose nucleic acid, so mRNA. And then once we have the conversion from DNA into mRNA, you can